Good morning everyone, we are group 7 and today we're going to present about the NMR spectrophotometer. And this is our table of content. We're going to talk about the introduction to NMR spectroscopy, the component and function, the principle and mechanism of NMR spectrophotometer, the sample preparation and handling, and the NMR parameter and NMR application. So what is NMR spectrophotometer? NMR is stand for the nuclear magnetic resonance, and the NMR spectrophotometry is an analytical technique that can detect the interaction of the radiofrequency electromagnetic radiation with the nucleus of the molecule placed in a strong magnetic field. It takes advantage of the magnetic properties of the nucleus to sense the chemical environment of the nucleus in a molecular structure. An NMR spectrophotometer can be operated in both liquid and solid state. And with the latest advancement, NMR spectrophotometer can also be utilized to investigate the suspension and colloidal nanomaterial. So next, we're going to talk about the component in the NMR spectrophotometer and its function. So first, it will have a permanent magnet which help in providing a homogeneous magnetic field at 60 to 100 MHz. And second one is the magnetic coil, which used to generate the magnetic field when current flows through it. And third one, it will have a magnet controller, which used to modify the strength of the magnetic field. The next one will be the radio frequency transmitter, which used to produce powerful but short pulse of the radio wave. And the fifth one is the radio frequency detector, which help in determining the unabsorbed radio frequency. The finally is the readout system, which is usually a computer that will analyze and record the data. So about the internal structure of the NMR spectrophotometer, it will have a cooling system inside. The magnet used in the NMR spectrophotometer are usually the superconducting materials, and thus they will require a very low temperature to work. Usually it's around 4 Kelvin. So the cooling system is actually composed of an inner jacket filled with the liquid helium, which is further refrigerated by an additional jacket filled with liquid nitrogen, and also many layers of thermal isolating materials.
To explain about the principles and mechanism of NMR spectrometer, we start with the nuclide it can detect. The NMR spectrometer can detect various nuclei with an odd number of neutrons which show magnetic properties. Common nuclei include proton, carbon-13, nitrogen-15, and fluorine-19, with protons and carbon-13 being the most widely used. NMR works because of the magnetic moment of certain nuclei. Therefore, nuclei with a quantum spin of 0 cannot be measured using NMR. This is because the spins of most isotopes with an even number of both protons and neutrons are paired and cancel each other out. They have no magnetic moment and therefore cannot be detected by an NMR spectrometer. Nuclei are positively charged and spin on the axis. This spinning creates a magnetic moment. When these nuclei are placed in an external magnetic field, the nuclear spins align either with or against the field, creating discrete energy levels. This alignment is crucial for the NMR process as it sets the stage for energy absorption and resonance, which we will discuss next. Energy absorption and resonance are key concepts in NMR. When the energy of the applied direction in the radio frequency RF range matches the energy difference between the nuclear spin, nuclei absorb energy and transition from the lower to the higher energy state. This condition is known as the resonance condition, and the corresponding frequency is called the Raman frequency. And NMR can differentiate between different elements and isotopes because each specific nuclide absorbs energy at a very specific frequency. Resonance occurs when the frequency of the applied RF radiation matches the Raman frequency of the nuclide in the magnetic field. Once nuclei are excited to a higher energy state, they eventually relax back to their lower energy state, emitting RF radiation in the process. And this emitted radiation is detected as the NMR signal, which is crucial for analyzing the sample. Let's look at how an NMR spectrometer works. First, the sample is placed in a magnetic field. The nuclei in a sample are then excited into resonance using radio wave producing NMR signals. These signals are detected by sensitive stereo receivers, and then by measuring the signal corresponding to this energy transfer, we obtain the NMR spectrum, which can be analyzed for the specific nucleus being studied. And the resonance frequency of an atom is affected by its intramolecular magnetic field, providing detailed information about the molecular structure, functional groups, and electronic environment. The position or chemical shift and the pattern of splitting of the NMR signals provide important information about the chemical environment of the nuclide. Chemical shift is the difference between the resonance frequency of the splitting nucleus and the signal of a reference molecule. It is expressed in parts per million ppm and provides valuable insights into the electronic structure surrounding the nuclide. This makes NMR spectrometer a powerful tool for determining molecular structures.
Hello everyone. Next, we will be discussing the crucial steps in sample preparation and handling for NMR spectroscopy. Proper preparation and handling ensures the accuracy and quality of your NMR spectra, which is essential for reliable results. In NMR, unlike other types of spectroscopy, the quality of the sample profoundly affects the quality of the resulting spectrum. Following a few simple rules can help you obtain clear and useful spectra. Number 1. Correct quantity of material. For proton spectra of organic compounds, use 5 to 25 milligrams of material. For C13 spectra, which are much less sensitive, prepare a saturated solution of about 0.2 to 0.3 millimole in 0.7 milliliters solvent. High concentrations can cause broader lines in proton spectra due to increased viscosity. Number 2. Removing solid particles. Solid particles can distort magnetic field homogeneity, causing broad lines and indistinct spectra. Filter all solutions into the NMR tube using a small plug of glass wool in a pasture pipette. Avoid using cotton wool as it can dissolve in solvents and contaminate your sample. Number 3. Correct sample depth. Prepare samples to a depth of 5 to 5.5 centimeters, requiring about 0.6 to 0.7 milliliters of solvent. This ensures proper magnetic field homogeneity and efficient shimming, which is crucial for obtaining high-quality spectra. Number 4. Use deuterated solvents. Deuterated solvents like CDCl3 provide a stable NMR lock signal. Use a calibrated all-glass syringe to handle these solvents to avoid contamination. Do not use them for solubility trials as they are costly. Number 5. Clean tubes and caps. Use clean 5 mm NMR tubes, rinse them with acetone, and dry with air or nitrogen. Avoid drying tubes in hot ovens as this can distort them. Always cap your tubes and ensure they are not chipped or broken. Number 6. Label samples. Label your samples with your user code and sample code using a solvent-based felt pen. Clean new tubes to remove any manufacturing grease. Number 7. Avoid adding TMS. Instead of adding TMS yourself, use solvents with pre-dissolved TMS if needed. Typically, the residual protons in deuterated solvents serve as a secondary reference. Number 8. Degassing samples. For degassing, use the freeze pump thaw technique for at least 3 cycles. Alternatively, flush the sample surface with nitrogen, being careful not to blow the solution out of the tube. Here are some tips to avoid contamination and degradation. Use clean equipment and wear gloves. Work in a clean environment. Control exposure to light, air, and moisture. Store samples properly to maintain their integrity. Proper sample preparation and handling are vital for achieving accurate and reliable NMR spectrometry results. By following these guidelines, we can ensure high quality spectra and meaningful data. Next, let's take a look at the parameters involved in NMR spectroscopy.
Now, let me introduce you to the NMR spectroscopy parameters. Firstly, the chemical shift. It is measured in parts per million ppm. It is the resonance frequency of a nucleus relative to a standard in a magnetic field. It reflects the electronic environment surrounding the nuclei, helping identify functional groups and elucidate molecular structures. Secondly, nuclear over Hauser effects, NOEs. NOEs are changes in NMR signal intensity caused by spatial proximity between nuclei through cross relaxation. NOEs provide information about spatial relationships between nuclei, particularly useful in NOESY to determine distance within molecules. Thirdly, diffusion constants. It measures how quickly molecules diffuse through a medium, determined using pulse field gradient PFG NMR. They reflect molecular size, shape, and interactions with the environment. And lastly, relaxation time T1 and T2. T1 is longitudinal relaxation time. It's the time taken for nuclei to return to thermal equilibrium along the magnetic field. It indicates molecular motion and environment affecting signal intensity and relaxation behavior. And lastly, transverse relaxation times T2 is the time taken for nuclei to lose phase coherence perpendicular to the magnetic field. It reflects molecular interactions and motions, providing insights into molecular rigidity and interactions. Next, let me show you the applications of NMR spectrophotometer in various fields. Firstly, in organic chemistry, NMR spectroscopy is crucial for identifying and elucidating the structure of organic compounds. It provides detailed information on hydrogen and carbon atoms, confirming the structure of synthesized molecules and natural products. It also verifies purity and composition and studies reaction mechanisms and kinetics. Secondly, in biochemistry, NMR is vital for studying the structure and dynamics of biomolecules like proteins, nucleic acids, and lipids. It determines their three-dimensional structures in solution, aiding in understanding their function and interactions. NMR profiles metabolites and studies enzyme kinetics and ligand binding, crucial for drug discovery and development. In material science, NMR characterizes the structure and dynamics of polymers, ceramics, and other materials at the molecular level. Solid-state NMR is particularly useful for non-crystalline materials, providing insight into properties like porosity, crystallinity, and molecular mobility. This is essential for developing materials with specific properties for applications in electronics, catalysis, and nanotechnology. Next, in medicines, NMR is widely used especially in magnetic resonance imaging, MRI. MRI visualizes soft tissues with high spatial resolutions, revolutionizing diagnostic for the brain, spine, joints, and other organs. In vivo, NMR studies metabolic changes in tissues, offering insights into conditions like cancer and neurological disorder. It also aids in drug development by analyzing the structure activity relationship of therapeutic agents. Next, in environmental science, NMR analyzes complex environmental samples like soils, sediments, and water, identifying pollutants and understanding their interactions with the environment. It studies natural organic matter and its role in carbon cycling and nutrient availability, crucial for pollution remediation and sustainable resources management. Next, in food science, NMR analyzes the compositions, quality, and authenticity of food products, identifying and quantifying components like fats, proteins, and carbohydrates. It detects adulteration and ensures compliance with food safety standards, and also studies the molecular interactions and dynamics in food, helping to understand texture, flavor, and shelf life. Lastly, in pharmaceuticals, NMR is vital for characterizing drug compounds and impurities, determine stereochemistry and polymorphism, and analyzing complex mixtures during drug development and quality control. It studies drug metabolism and pharmacokinetics, providing valuable information on how drugs are absorbed, distributed, metabolized, and excreted. Here is all the applications of NMR spectrophotometer in various fields. In conclusion, NMR, which is nuclear magnetic resonance spectrophotometers, are essential in biotechnology research, providing detailed insights into biomolecular structures, dynamics, and functions. Their high resolution, non destructive analysis enables the study of proteins, nucleic acids, and metabolites, facilitating breakthroughs in drug discovery, structural biology, and metabolic engineering.
So these are the list of references that we've used for our presentation, which marks the end of our video. Thank you for watching and please leave your questions in the comment section.